Autophagy plays a crucial and often underappreciated role in protecting mitochondrial health, especially in the context of obesity, where energy systems are chronically strained and frequently dysfunctional. Among the various forms of autophagy, mitophagy, the targeted removal of damaged or dysfunctional mitochondria, is particularly important. Mitochondria are the powerhouses of the cell, responsible for generating the energy needed for all metabolic activity, including fat oxidation, glucose metabolism, and thermogenesis. When mitochondrial quality control breaks down, the body becomes less efficient at using energy, more prone to fat storage, and more vulnerable to metabolic disorders like insulin resistance, inflammation, and type 2 diabetes. In healthy individuals, mitophagy ensures that only fully functional mitochondria remain. This not only optimizes energy production but also prevents the release of harmful byproducts, such as reactive oxygen species, RLS, which are known to damage cellular components and trigger inflammatory signaling. Obesity, however, increases the burden on mitochondria due to excess nutrient intake, chronic oxidative stress, and persistent low-grade inflammation. Over time, this leads to a buildup of dysfunctional mitochondria, which can no longer produce ADP efficiently and instead leak damaging compounds that further impair metabolic tissues. This is where autophagy becomes vital. By identifying and engulfing damaged mitochondria, mitophagy prevents their harmful effects and creates space for the production of new, healthier mitochondria, a process known as mitochondrial biogenesis. This turnover is essential in metabolically active tissues like skeletal muscle, liver, heart, and even in fat cells, where mitochondrial dysfunction can directly impair the ability to burn fat or regulate glucose. Unfortunately, in many obese individuals, autophagic activity, especially mitophagy, is suppressed. This means that damaged mitochondria are not effectively cleared, and their accumulation leads to a cascade of metabolic dysfunction. For instance, in skeletal muscle, defective mitophagy has been linked to poor glucose uptake, muscle fatigue, and insulin resistance. In the liver, it contributes to steatosis and impaired gluconeogenesis. Even in brown fat, which is designed to burn calories through thermogenesis, poor mitophagy leads to reduced energy expenditure and worsening fat accumulation. Thus, mitophagy failure is both a consequence and a driver of the metabolic chaos seen in obesity. Yet, despite these risks, there is reason for optimism. Mitophagy is not a fixed process. It can be reactivated. Interventions such as endurance exercise, caloric restriction, intermittent fasting, and certain plant compounds, like urolithin A, resveratrol, or quercetin, have been shown to stimulate mitophagy and improve mitochondrial quality. When mitophagy is enhanced, cells regain their capacity to efficiently use fuel, regulate energy, and resist oxidative damage. In animal models and emerging human studies, these interventions not only reduce fat mass, but also improve glucose metabolism and reduce inflammatory markers. It is worth noting that mitochondrial quality is more important than quantity. Simply having more mitochondria is not beneficial if they are inefficient or damaged. Mitophagy ensures that a smaller pool of high-performing mitochondria can better support energy metabolism than a larger group of malfunctioning ones. This insight changes the focus from simply boosting metabolic rate to optimizing the machinery behind it. There's also growing interest in the possibility that early enhancement of mitophagy could serve as a preventive strategy, especially for individuals with a genetic predisposition to obesity or those exposed to high-fat, high-sugar diets from a young age. By preserving mitochondrial health early on, it may be possible to maintain metabolic flexibility, resist weight gain, and prevent the progression to full-blown metabolic disease. In summary, autophagy, through its specialized form of mitophagy, serves as a cellular quality control mechanism that is essential for maintaining mitochondrial function and energy homeostasis. When this system breaks down in obesity, cells become less efficient, more inflamed, and increasingly dysfunctional. But this dysfunction is not permanent. By reactivating mitophagy through lifestyle and nutritional strategies, it is possible to restore metabolic resilience, burn fat more efficiently, and slow or reverse many of the complications associated with obesity. Mitochondrial health is not a fixed trait, it is maintained by ongoing surveillance, and autophagy is the key to that maintenance.